Welcome back to the Deep Dive. Today we're opening a file that I think is one of the most compelling examples of how tiny, almost imperceptible differences in biology can lead to radically different destinies.
It's a fascinating case study.
We're looking at a pair of siblings in a bacterial world. If you look at them under a microscope, they are, for all intents and purposes, twins. They're visually indistinguishable.
Completely.
But their behavior, their lifestyle choices, if you will, it couldn't be more different.
It really is a Prince and the Pauper situation, or maybe more accurately, a Dr. Jekyll and Mr. Hyde scenario, but split between two different organisms.
That's a great way to put it.
On one side, you have a pathogen that is a master of local chaos. It causes messy, painful mucosal infections, but it rarely kills you.
Right.
On the other side, you have a sibling that can live quietly in your throat for months, doing absolutely nothing until one day it just snaps. It invades your bloodstream, and it can end a life in less than 24 hours.
We are talking, of course, about the genus Neisseria. Specifically, the two major human pathogens, Neisseria gonorrhoeae and Neisseria meningitidis.
The gonococcus and the meningococcus.
And to guide us through this complex family, we're using the structural spine of Murray's Medical Microbiology, Chapter 23. We'll also be pulling in some key supplementary details from Japheth's, Chapter 20.
Right, and while these are, heavily featured on medical board exams, our goal today isn't just to memorize facts. We really want to understand the why.
The mechanism.
Exactly. Why does one stay local and the other go systemic? Why do we have a vaccine for one but not the other? It all comes down to their molecular architecture.
So let's start with that visual identity. If I'm a student looking at a gram stain from a clinical specimen, let's say it's purulent discharge or maybe spinal fluid, what is the image that should just scream Neisseria?
It is the gram-negative diplococcus, so pink or red pairs. But the shape is very, very specific. They aren't perfect spheres like, say, staphylococcus.
They're a bit squashed.
A bit squashed and always in pairs. The classic description is kidney beans or coffee beans.
Okay, I can picture that.
And they are always oriented in a specific way. The flat or concave sides are adjacent. So if you imagine two kidney beans, they're sort of facing each other with their flat sides kissing.
So they're not end-to-end like some other bacteria?
No, never. And context is everything here. In a real clinical sample, you are very often seeing these pairs inside a polymorphonuclear leukocyte, a PMN.
A neutrophil.
A neutrophil, exactly.
Which is a really powerful image when you think about it. You see this massive immune cell, this professional killer, and inside its cytoplasm, you see these little red pairs of bacteria.
Yeah.
Your first thought is, great, the immune system is doing its job. It's eating the invaders.
But as we're going to find out later, eating them doesn't necessarily mean killing them. In fact, for Neisseria, it can be part of the plan.
A survival strategy.
Precisely. That intracellular finding is a hallmark of the genus, a high-yield image for any exam.
Okay, so before we get to the crimes of these bacteria, let's understand their basic physiology. What makes a Neisseria? Murray calls this the shared chassis, which I think is a great term.
It is. These are the fundamental traits that both the gonococcus and the meningococcus share.
And the first one is a big one in the lab.
First and foremost, they are oxidase positive.
A purple ring test.
Exactly. It's a biochemical test where we look for the presence of an enzyme, cytochrome C oxidase, which is part of their respiratory chain. You smear a colony on filter paper with a specific reagent, and if the enzyme is there, it turns a dark purple or black within seconds.
And why is that so important?
Because it's a major fork in the road for identifying gram-negative bacteria. It cleanly separates them from the massive family of Enterobacteriaceae, the common gut bacteria like E. coli, Klebsiella, Proteus. All of those are oxidase negative.
So you see a gram-negative diplococcus, you do an oxidase test, it turns purple. You are now strongly suspecting Neisseria.
You are. They're also catalase positive, which means they can break down hydrogen peroxide. But the really distinct feature is their metabolism. They oxidize carbohydrates.
They don't ferment them.
They do not ferment them. This is a crucial distinction. Many of those gut bacteria we mentioned are fermenters. They produce large amounts of strong acid from sugars, often with gas. Neisseria are more delicate. They oxidize sugars, producing a smaller amount of weak acid.
And this difference in metabolism brings us to probably the most famous differentiator in all of microbiology. If they look identical under the microscope, how do we tell the twins apart in the lab? It comes down to what they eat, right?
It does. It's their sugar preference. We use a specific type of media, usually cysteine trypticase agar, or CTA, that's supplemented with a single carbohydrate and a pH indicator.
So you'll have one tube with glucose, one with maltose, maybe one with sucrose.
Exactly. And the pattern is the most classic mnemonic in the field. Let's break it down.
Let's do it.
Neisseria gonorrhoeae produces acid from glucose only, just glucose. It can't touch maltose, lactose, or sucrose.
So the mnemonic is G is for gonorrhea, and G is for glucose,
Simple as that. It only eats glucose.
and the meningococcus.
Neisseria meningitidis is a bit more versatile. It utilizes both glucose A and D maltose.
So M is for meningitis, and M is for maltose.
Precisely. If you inoculate both bacteria into a set of sugar tubes, the gonococcus tube will only turn yellow, indicating acid in the glucose tube. The meningococcus will turn yellow in both the glucose and the maltose tubes.
That's such a clean, elegant distinction. It seems simple, but it reflects a fundamental enzymatic difference between them.
It absolutely does.
Now, I want to talk about their survival needs. Reading through Murray and Jowett's, these bacteria seem incredibly high maintenance. The word that keeps coming up is fastidious.
Fastidious is the polite microbiological term for diva. They are incredibly fussy eaters, and they're very sensitive about their environment. You can't just grow them on standard nutrient agar.
They just die.
They would. They need an enriched medium, like chocolate agar, which is essentially cooked blood that releases growth factors, and they have very specific demands.
Okay, what specifically are they demanding on their writer, so to speak?
First and foremost, they require iron. But here's the fascinating evolutionary twist. Most bacteria produce things called siderophores.
Right, the little scavenger molecules.
Yes, they send these out into the environment to find and grab onto any free iron and bring it back. Neisseria don't do that.
Too much work.
It seems so. Instead, they have evolved highly specific receptors on their outer membrane that can bind directly to human iron-binding proteins.
So they're not looking for loose change on the floor.
No, they're picking the pocket of the host directly. They have receptors for human transferrin, lactoferrin, even hemoglobin.
That feels like a very bold, very aggressive strategy.
It is bold, but it comes with a significant cost. Because these receptors are exquisitely specific for human transferrin and lactoferrin, Neisseria are strict human pathogens.
Meaning they can't infect other animals.
Correct. You can't give a mouse or a dog gonorrhea because their iron-stealing keys don't fit those animal locks. This makes research very difficult because you can't easily study the diseases in animal models, but it tells us that these bacteria have co-evolved intimately with us for a very long time.
And it's not just their diet, they're also sensitive to the physical environment, right?
Extremely. They need a body temperature around 35 to 37 degrees Celsius. They need a humid atmosphere. And crucially, they require an atmosphere supplemented with carbon dioxide, usually about 5% CO2.
Like a captophile.
Yes. And they are incredibly fragile. If you let a clinical sample dry out on a swab, the bacteria die. If you put the sample in the refrigerator, they die from the cold shock.
This has huge implications for transmission and really debunks a lot of the common myths.
Absolutely. You cannot get gonorrhea from a toilet seat or a doorknob or a swimming pool. The bacteria simply cannot survive the cold, dry, oxygen-rich environment outside the human body for more than a few moments.
So transmission requires direct mucosal contact.
It has to be intimate, direct contact between mucosal surfaces. That's the only way they can survive the journey.
Okay, that gives us a solid foundation for the family. Let's separate the twins now and dive into their individual stories. We'll start with sibling number 1, Neisseria gonorrhoeae, the gonococcus.
The party animal.
Exactly. This is the one known for causing widespread, sexually transmitted infections and a whole lot of local misery. And structurally, the first and most important feature of the gonococcus is actually what it lacks.
It does not have a polysaccharide capsule.
It's the armorless warrior.
Perfect analogy. And this is going to be the single biggest point of contrast with meningococcus later. The surface of gonococcus has a slight negative charge, which helps repel phagocytes a bit, but it doesn't have that thick, tank-like carbohydrate armor.
So without heavy armor, it has to rely on other strategies, stealth, adhesion, misdirection.
Exactly. It has a whole toolbox of virulence factors designed for a life of mucosal guerrilla warfare.
Let's unpack that toolkit.
Yeah.
The first and maybe most famous tool is the pili.
The pili are these long, hair-like protein appendages that extend from the bacterial surface. Their primary job is attachment.
Grappling hooks.
They are absolutely like grappling hooks. They allow the bacteria to make that initial contact and latch onto non-ciliated epithelial cells, like the ones in the urethra, cervix, fallopian tube, or rectum.
But the pilis are so much more than just simple hooks. This is where the master of disguise element comes into play.
This is one of the most sophisticated and clinically important mechanisms in all of bacteriology. The immune system is constantly looking for these pili. If it sees them, it makes antibodies to coat them, block their function, and tag them for destruction.
A very standard immune response.
A very standard response that Neisseria gonorrhoeae is completely prepared for. It uses a process called antigenic variation to constantly change what the pili look like.
I really want to go deep on this. How exactly does it change its face? Is it just a high rate of random mutation?
No, it's much more elegant and organized than that. It's a pre-programmed genetic shuffling mechanism. So think of it this way. The bacterium has one active gene locus called pile, where the gene for the main pilin protein subunit is located and actively being expressed.
That's the gene that's currently making the grappling hooks.
Correct. But elsewhere in its chromosome, it has a whole library of silent, non-expressed, partial copies of the gene. These are called piles cassettes.
So pilis are like the backup wardrobe, a whole closet of different coats.
That is a perfect analogy. These silent piles copies are all slightly different from each other, especially in the regions that code for the exposed antigenic parts of the protein.
The parts the antibodies see.
Exactly. Through a process of homologous recombination, the bacteria can take a piece of one of the silent pilis genes and swap it into the active pile expression site.
It's like a card shark swapping an ace from his sleeve into his hand.
It is exactly like that. The result is that the bacteria suddenly starts producing a new version of the pilin protein. The antibody you made last week against version A is now completely useless against version B, which is being made today.
And this happens constantly within a single infection.
Constantly. It's happening all the time, allowing the bacterial population to stay one step ahead of the host's adaptive immune response.
And this directly explains two huge clinical problems. First, why we have never been able to make a vaccine for gonorrhea.
Correct. A vaccine relies on teaching the immune system to recognize a stable target. If the target is a shapeshifter, the vaccine is doomed to fail.
And second, it explains why you can get gonorrhea over and over again.
Yes. There is no lasting protective immunity. An infection this month provides no real protection against an infection 6 months from now because the new strain you encounter will likely have a different pilin code.
That is incredibly frustrating for public health. But from a biological standpoint, it is brilliant for the bacteria.
It is a masterclass in immune evasion.
Okay, what else is in the toolbox? Pilis are for initial attachment. What about staying put and surviving the onslaught?
Let's talk about the pore proteins, specifically PORB. These are porin proteins, which are basically channels or pores in the outer membrane that let nutrients in and waste out. But for gonorrhea, PORB has been co-opted into a defensive weapon.
How does a nutrient channel act as a weapon? That seems counterintuitive.
It works by interfering with the neutrophil's digestion process. Remember we said the bacteria often end up inside neutrophils?
Right.
Normally, after a neutrophil engulfs a bacterium, it fuses that bacteria-containing vacuole, the phagosome, with another compartment filled with acid and digestive enzymes, the lysosome.
Phagolysosome fusion. That's the kill step.
That is the kill step. PORB actually prevents this fusion. It translocates to the phagosome membrane and sends a signal that stops the lysosome from docking and fusing.
So the bacteria just sits comfortably inside the phagosome, protected from the acid bath.
Exactly. It's protected from the outside world by the neutrophil's own membrane and protected from the neutrophil's weapons by PORB. It can even replicate inside the white blood cell.
So when we see that classic gram stain image of the neutrophil full of diplococci, the neutrophil isn't winning. It's being used as a hotel. Or a Trojan horse.
In many cases, yes. The bacteria are using the host's own primary defense cells as a shield to hide from antibodies and even some antibiotics.
That's incredible. There are two other key virulence factors Murray mentions that I want to touch on. OPA proteins and LOS.
OPA proteins stand for opacity proteins. They get their name because when a colony on an agar plate expresses them, it looks opaque or cloudy. When it doesn't, it looks translucent.
But what do they do biologically?
They are critical for firm attachment. If pili are the grappling hooks for getting close, OPA proteins are like the superglue for sticking tightly to the host cell. They also mediate binding between one gonococcus and another.
Why is that important?
It's critical for forming microcolonies and biofilms on the mucosal surface. And interestingly, there's a clinical correlation. Isolates from localized infections, like a simple urethritis, tend to be OPA positive. They want to stick around.
But if the infection disseminates,
Isolates from disseminated infections often switch off OPA expression. They become transparent. The theory is that this makes them less sticky, more slippery, and better able to detach and travel through the bloodstream.
and finally LOS. We almost always talk about LPS, lipopolysaccharide, in gram negatives. Why the different letter?
It's a structural difference. Neisseria produces lipooligosaccharide, LOS. It has the two core components of LPS lipid A, which is the toxic part, and a core oligosaccharide. But it lacks the long, repetitive O antigen chain that you see in things like E. coli or Salmonella.
So it's like a short, stubby version of LPS.
Exactly, but don't let the oligo, meaning few, fool you. This molecule is a potent endotoxin. During rapid growth, the bacteria are constantly shedding or blebbing off bits of their outer membrane that are packed with LOS.
And blebbing is a great word, but the effect is nasty.
It's the primary driver of inflammation in gonorrhea. The lipid A portion of LOS binds to toll-like receptors on immune cells and triggers a massive release of inflammatory cytokines, especially TNF-alpha.
Tumor necrosis factor alpha.
Right, and TNF-alpha is what causes the local damage, the pain, the redness, the influx of neutrophils that create the pus, and very importantly, the destruction of the mucosal cilia.
Which brings us directly to the clinical picture. Let's walk through the disease manifestation because it's so different between men and women.
It really is. It starts with exposure, and the incubation period is quite short, usually 2 to 5 days. In men, the symptoms are typically acute and obvious.
Hard to ignore.
Very hard to ignore. It presents as urethritis. The patient experiences dysuria, a burning, painful sensation during urination, and a purulent, thick, yellow-green discharge from the urethra.
The classic drip.
Exactly. And because it's so painful and so visual, about 95% of infected men are symptomatic and seek treatment early. This generally limits the long-term complications in men.
But in women, the story is dangerously different.
It is. In women, the primary site of infection is the endocervix. And the tragedy here is that up to 50% of infected women have very mild symptoms or are completely asymptomatic.
So they might have a little bit of vaginal discharge, some minor dysuria.
But they often mistake it for a common yeast infection or a UTI. They don't recognize it as a serious STI.
So they don't seek treatment. And this has two major consequences.
Two huge ones. First, they become the major reservoir for transmission in the community. And second, and more importantly for their own health, the bacteria have time to ascend.
Go exploring.
They move from the cervix up through the uterus and into the fallopian tubes. This leads to pelvic inflammatory disease, or PID.
Yeah, PID is a disaster for reproductive health.
An absolute disaster. That intense inflammation caused by the LOS endotoxin essentially destroys the delicate ciliated cells that line the fallopian tubes.
And those cilia are needed to gently sweep the egg from the ovary to the uterus.
Correct. So if you scar those tubes shut, you get infertility. Or if the tube is only partially blocked and scarred, the egg might get fertilized, but then get stuck on its way to the uterus.
An ectopic pregnancy. A life-threatening emergency.
Yes. And all of this is a silent consequence of an infection that may have caused no initial symptoms at all.
There's a specific syndrome associated with this ascent that comes up in exams and clinical rounds constantly. Fitzhugh-Curtis syndrome.
This is perihepatitis. So the infection can spill out of the open ends of the fallopian tubes into the peritoneal cavity and cause inflammation of the capsule of the liver, which is called Glisson's capsule.
And the visual finding during surgery is iconic.
It is. Surgeons see these thin, fibrous adhesions stretching from the anterior surface of the liver to the abdominal wall. They look just like the strings of a violin.
Violin string adhesions.
That's the one. And clinically, this presents as right upper quadrant pain in a sexually active young woman. It can easily mimic gallbladder disease or cholecystitis, but the underlying cause is gonorrhea.
Now, for the most part, gonorrhea stays local. But in about 1 to 3% of cases, it decides to go rogue. It gets into the bloodstream.
This is disseminated gonococcal infection, or DGI.
And it's more common in women.
Much more common in women. And the reason is that untreated asymptomatic reservoir. If the infection smolders for weeks or months, there's just more opportunity for it to breach into the blood. Menstruation also seems to be a risk factor for dissemination.
And DGI has a very specific triad of symptoms that you need to recognize.
Yes. The first is migratory polyarthralgia. This is joint pain that moves around. Today my wrist hurts, tomorrow it's my knee, the next day it's my ankle. The joints are painful, but not usually hot and swollen.
Okay, what's number 2?
Number 2 is tendinovitis. This is inflammation of the tendon sheaths, especially in the hands and feet. So you get pain with movement of the fingers or toes.
And the third part of the triad.
Dermatitis, a skin rash. But it's not a widespread rash like you see in measles. It's typically a few, sometimes just 5 or 10 pustules on an erythematous base, and they're most common on the extremities, not the trunk.
If that infection decides to settle down on one of those painful joints, it can become a true purulent arthritis.
Yes, and this is a major clinical point. Neisseria gonorrhoeae is the leading cause of septic arthritis in young, sexually active adults. If a 25-year-old comes into the emergency room with a single, hot, swollen, painful, unmovable knee, you have to suspect DGI.
I have to tap that joint.
You have to aspirate the fluid and send it for gram stain and culture immediately.
Before we move to diagnosis, we have to mention the babies. Ophthalmia neonatorum.
A heartbreaking but largely preventable condition. If a mother has an untreated gonococcal infection of the cervix, the baby is exposed to the bacteria during passage through the birth canal.
And their eyes are vulnerable.
Very. It causes a severe, hyperacute, purulent conjunctivitis, usually within the first 5 days of life. The eyelids are swollen shut, and there's a massive amount of pus. If it's not treated aggressively, it can lead to corneal ulceration and permanent blindness very, very quickly.
And this is precisely why we have the universal eye ointment.
Exactly. In most developed countries, a prophylactic ointment, usually erythromycin, is applied to essentially all newborns' eyes immediately after birth. It's a simple, cheap public health triumph that has made gonococcal blindness a rarity.
Okay, let's talk about catching the culprit. Diagnosis.
It depends heavily on the patient. For a man with symptomatic urethritis, the simple gram stain is excellent. You take a sample of the discharge, stain it, and look under the microscope.
And you see the kidney beans inside the neutrophils.
You do. In that specific context, it's more than 90% sensitive and very specific. You can make a presumptive diagnosis and treat based on the gram stain alone.
But for women, it's not so simple.
No. For a cervical swab from a woman, the sensitivity of the gram stain plummets to about 50%. The female genital tract has a lot of normal commensal flora, including other gram-negative bacteria like Acinetobacter or Moraxella that can look similar.
So for women or for asymptomatic men or for samples from the rectum or pharynx, you need to do more.
You must. You need to confirm with either culture or a molecular test.
But culturing this diva isn't easy, especially if you're taking a sample from a non-sterile site like the cervix or rectum. There are millions of other bacteria there that will outgrow the Neisseria.
You need a way to kill the competition while letting the Neisseria grow. And this brings us to the famous Thayer-Martin agar.
The bouncer of the petri dish.
It's the perfect description. It starts as chocolate agar, which is the rich medium the bacteria needs. But then it's supplemented with a very specific cocktail of antibiotics, often called VCN.
Let's break that down.
V for vancomycin. This kills most of the gram-positive organisms like staph and strep. C for colistin. This is an antibiotic that kills most other gram-negative organisms, especially the enteric rods like E. coli.
Fin.
N for nystatin. This is an antifungal agent to kill any yeast, like Candida, that might be present.
So if something manages to grow on Thayer-Martin agar, it has survived the vancomycin, the colistin, and the nystatin. It is almost certainly a pathogenic Neisseria.
Exactly. It's a highly selective medium. Though I should say, today in most clinics, the gold standard has become nucleic acid amplification tests, or NATs.
TCR tests.
Right. They're incredibly sensitive and specific. They give you a result in hours instead of days, and they can often be done on a simple urine sample, avoiding the need for an invasive swab.
Okay, let's wrap up the gonococcus with treatment. And this, unfortunately, is the doom chapter of the story.
It is a genuine public health crisis. Neisseria gonorrhoeae is incredibly adept at acquiring antibiotic resistance. It's a story of one fallen drug after another.
Penicillin used to work.
It did, until the bacteria picked up plasmids that carried a gene for penicillinase, an enzyme that just chews up penicillin. So penicillin failed.
Then we used tetracyclines.
And the bacteria mutated its ribosomes so the tetracycline couldn't bind anymore. So tetracyclines failed.
Then the fluoroquinolones, like Cipro.
Widespread resistance developed through mutations in the DNA gyrase enzyme, the target of the fluoroquinolones. They failed too.
So it has systematically defeated almost every major antibiotic class we've thrown at it.
We are down to our last reliably effective options. The current CDC recommendation is ceftriaxone, which is a third-generation cephalosporin given as a single intramuscular injection.
But it's not given alone anymore.
No. For a long time it was, but now it's given as dual therapy with azithromycin, which is given orally.
Why the dual therapy? Is it just to hit it harder?
There are a couple of reasons. One is a practical one, to provide coverage for chlamydia, which is a very common co-infection with gonorrhea. But the main reason, from a resistance standpoint, is to protect the ceftriaxone.
How does that work?
By using two drugs with completely different mechanisms of action, you make it much harder for the bacteria to survive. It would have to develop resistance to both drugs simultaneously, which is statistically much less likely. It's a strategy to preserve our last best drug.
But we are seeing strains with reduced susceptibility even to ceftriaxone.
We are. The specter of untreatable gonorrhea is very real. We are running out of drugs.
That is a very sobering thought. Okay, let's pivot. We're leaving the party animal and moving to the quiet killer. Sibling number 2, Neisseria meningitidis, the meningococcus.
And structurally, there is one massive, overwhelming difference between this organism and its brother.
The polysaccharide capsule.
Yes. If the gonococcus is a nimble ninja relying on stealth and evasion, the meningococcus is a heavily armored tank.
A tank is the perfect metaphor.
This capsule is a thick layer of sugar molecules that wraps the entire bacterium. It is the single most important virulence factor for this organism.
And what does it do?
Its primary function is to be antiphagocytic. It's incredibly slippery, and it masks the underlying bacterial components from the immune system. A neutrophil literally can't get a grip on it. It also helps it resist killing by the complement system.
So it's a shield that allows the bacteria to survive in the bloodstream.
Exactly. The bloodstream is normally a death zone for most bacteria. The capsule is what allows the meningococcus to thrive there.
And we classify the different strains of meningococcus based on the chemical composition of this capsule, right?
Yes. There are 13 known serogroups based on the capsule structure, but almost all human disease is caused by 6 of them: A, B, C, Y, W135, and X. Knowing the serogroup is absolutely critical for epidemiology and for vaccine purposes.
Let's trace the journey of the pathogen here, because unlike gonorrhea, this infection usually starts with nothing.
It starts with asymptomatic colonization. The bacteria is transmitted person to person via respiratory droplets, coughing, sneezing, kissing, sharing drinks in a crowded setting.
So it lands in the nasopharynx, the back of the throat.
Correct. And just like gonorrhea, it uses its palatica to attach to the non-ciliated epithelial cells in the throat.
And for most people, that's where the story ends.
That is exactly right. At any given time, 10% to 20% of the healthy population are asymptomatic carriers of Neisseria meningitidis. It's just living in their throat, their immune system is keeping it in check, and it's causing no harm whatsoever. This is the carrier state.
But sometimes, for reasons we don't fully understand, the tank starts rolling.
Yes. In a small percentage of susceptible individuals, maybe someone who lacks specific antibodies or their throat mucosa is irritated from a recent viral infection or from smoking, the bacteria manage to penetrate the epithelial lining and enter the bloodstream.
This is the invasion phase, and now they are in the blood. This is bacteremia.
And because of that antiphagocytic capsule, the immune system struggles to clear them. They begin to replicate, and they can replicate very, very rapidly. And as they replicate, they shed massive amounts of that LOS endotoxin we talked about.
And this is where the quiet drifter becomes the arsonist who burns the whole house down.
The level of endotoxin, or LOS, in the blood of a patient with severe meningococcaemia can be 100 to 1,000 times higher than what you see in other types of bacterial sepsis.
That's an incredible amount.
It is. And it triggers a massive, uncontrolled systemic immune response. A cytokine storm, TNF-alpha, interleukins, they all flood the system.
And this leads to catastrophic systemic effects.
The cytokines cause the capillaries all over the body to become leaky, plasma pours out into the tissues, blood pressure plummets, and you go into shock. At the same time, the clotting cascade is activated everywhere at once.
Disseminated intravascular coagulation, or DIC.
Exactly. You start forming tiny clots in small blood vessels, which cuts off blood flow to the fingers, toes, skin, and vital organs. Then you use up all your clotting factors, and you start bleeding uncontrollably from everywhere else. It's a complete circulatory collapse.
Let's connect this pathophysiology to the clinical presentation. We should probably separate meningitis from meningococcaemia.
We should. Meningitis is when the bacteria cross the blood-brain barrier and infect the meninges, the lining of the brain and spinal cord.
And this causes the classic signs.
Yes. The abrupt onset of a high fever, a terrible, unrelenting headache, a stiff neck, what we call neutral rigidity, and photophobia, a painful sensitivity to light.
But meningococcaemia, the sepsis syndrome, has a specific sign that every medical student and clinician is trained to spot on site.
The petechial rash. These are tiny, pinpoint red or purple spots on the skin. They're not like a normal rash. They are actually small hemorrhages from blood leaking out of those damaged capillaries.
And they can grow larger.
They can coalesce into larger bruise-like areas called purpura or ecchymosis. The key diagnostic test you can do at the bedside is the glass test or tumbler test.
Describe that.
You press a clear glass firmly against the rash. If the spots disappear or fade under the pressure, they blanch, which is reassuring. But if the spots do not fade, that means the blood is outside the vessel. It's a non-blanching rash.
And what does that signify?
Fever plus a non-blanching petechial rash is meningococcaemia until proven otherwise. It is a true medical emergency. You must start antibiotics immediately. Minutes matter.
And the most severe, fulminant form of this sepsis has a name that sounds like a thunderclap. Waterhouse-Friderichsen syndrome.
This is the absolute nightmare scenario. It's the end stage of that disseminated clotting and bleeding we talked about, and it specifically hits the adrenal glands.
Why the adrenals?
The adrenal glands are incredibly vascular. They have a very rich blood supply. And when you have this widespread endothelial damage and clotting, you get bilateral hemorrhage into the adrenal glands. They essentially turn into bags of blood and are completely destroyed.
So in an instant, you lose your ability to make adrenaline and cortisol.
Acute adrenal insufficiency. Right at the moment when your body is under the maximum possible stress from septic shock and desperately needs cortisol to maintain blood pressure, the factory that makes it burns down.
And the result is?
Profound refractory shock, complete cardiovascular collapse, and very rapid death. It's terrifying how quickly this can happen. A healthy teenager can go from feeling a bit unwell to being dead in less than 12 hours.
Now, Murray's chapter mentions a very specific host risk factor that dramatically increases the risk for this kind of invasive Neisseria disease.
Yes. We've talked about how the bacteria fights the immune system, but one of the host's main weapons is the complement system, specifically a part of it called the membrane attack complex, or MA.
It's like a molecular drill.
That's a great way to think of it. It's a group of proteins, C5b through C9, that assemble on the surface of a bacterium and literally punch a hole through its membrane, causing it to lyse and die.
And this MAC is particularly important for killing Neisseria.
It is absolutely essential. Patients who have a genetic deficiency in any of the late components of complement, that's C5, C6, C7, C8, or C9, cannot form this drill.
And what's the consequence?
They have a 6,000-fold increased risk of invasive meningococcal disease. It's a massive risk factor. Any patient who has had recurrent meningococcal or even disseminated gonococcal infections should be screened for a complement deficiency.
Let's shift to the good news. Unlike gonorrhea, we have vaccines for the tank. But the story of the vaccine development is a fascinating lesson in biochemistry and immunology.
It really is. We have very effective vaccines against the major serogroups A, C, Y, and W135. The most common ones given to adolescents are the conjugate vaccines, like Men-S-WY.
Okay, let's break down the word conjugate. Why can't we just inject the purified capsule sugar itself?
Because polysaccharides, long chains of sugar, are what we call T-cell independent antigens. They can stimulate B cells to make some antibody, but the response is weak, short-lived, and doesn't generate good immune memory. It's especially poor in infants and young children.
So how does conjugation fix that?
The conjugate trick is to chemically link or conjugate the polysaccharide to a protein carrier that the immune system recognizes really well, like a non-toxic version of the diphtheria toxin.
The protein acts like a beacon for the T cells.
Exactly. The B cell recognizes the sugar, but it also presents the attached protein to a T helper cell. The T cell then provides a powerful activation signal back to the B cell, telling it to switch on, multiply, and make massive amounts of high-affinity antibody against the sugar.
So it turns a weak T-independent response into a strong T-dependent response with memory.
Precisely. It's a clever way to trick the immune system into making a much better response to the sugar.
But for the longest time, there was a glaring hole in this shield. We had no vaccine for serogroup B, and serogroup B is a major cause of meningitis in Europe and the Americas. Why was B so difficult?
This is a beautiful and slightly terrifying case of molecular mimicry. The capsule of serogroup B is made of a sugar called polysalic acid.
Okay.
It turns out that polysalic acid is chemically identical to a carbohydrate molecule called NCAM, the neural cell adhesion molecule, which is found on the surface of human fetal neural tissues.
So the bacteria is wearing a coat made out of a material that looks like human baby brain.
That's exactly right. Our immune system is trained from birth not to attack our own tissues. This is called self-tolerance. So when it sees the serogroup B capsule, it recognizes it as self and ignores it.
And if you tried to make a vaccine out of it?
You'd risk breaking that tolerance and inducing an autoimmune response against our own nervous system. So for decades, we couldn't use the capsular approach for serogroup B.
So how did we finally solve it? Because we do have Men-B vaccines now.
We solved it using a brilliant approach called reverse vaccinology.
What does that mean?
Instead of starting with the live bacteria and trying to find things on its surface, scientists started with the DNA. They sequenced the entire genome of a Neisseria meningitidis serogroup B strain.
They read the complete instruction manual.
Right. Then they used computer algorithms to scan the entire genome and predict which genes coded for proteins that would likely be on the outer surface of the bacteria, and crucially, were not similar to any known human proteins.
They were looking for unique, non-human targets.
Exactly. They identified several promising candidates, like factor H binding protein or FHBP, and manufactured these proteins. These are the basis for the modern Men-B vaccines like Bexsero and Trumanba. They target proteins on the tank, not the armor itself. It was a triumph of genomic science.
What about treatment for an active infection?
It is a medical emergency. You do not wait for the lumbar puncture results if it will delay starting antibiotics. You start treatment immediately on suspicion.
And the drug of choice?
A third-generation cephalosporin, like ceftriaxone or cefotaxime. They have excellent activity against Neisseria, and they penetrate the blood-brain barrier very well.
And what about the people who have been in close contact? If my college dorm mate gets diagnosed with meningitis, what happens to me?
You and anyone else with close contact need chemoprophylaxis. The goal is to eradicate any carrier state in your nasopharynx before it has a chance to invade.
And what drugs do we use for that?
Rifampin is the classic drug, given for 2 days. Ciprofloxacin as a single dose is also effective, or a single injection of ceftriaxone.
Rifampin is the one with a famous colorful side effect, isn't it?
It is. It turns all body fluids, urine, tears, sweat, a bright orange red. If you wear soft contact lenses, they will be permanently stained orange. You absolutely have to warn the patient about this.
Good to know. Okay, we have covered the two giants of the family, but Murray's chapter also mentions some related genera. These are the cousins. They aren't as famous, but they have very specific clinical niches and exam buzzwords.
Let's start with Eikenella corrodens.
I call this the fifth fight bacteria.
That is the perfect association for it. Eikenella is part of the normal flora in the human mouth. It's a HA-seq organism. It doesn't really cause pharyngitis, but it causes huge problems when it gets inoculated into deep tissue.
And the most common way for that to happen is either a direct human bite or the so-called clenched fist injury.
Exactly. Someone gets angry, they punch someone else in the mouth. The victim's teeth then act like little scalpels, shattering the skin over the aggressor's knuckles.
And the bacteria from the mouth get driven deep into the tendon sheaths and joints of the hand.
It creates a nasty polymicrobial infection, and Eikenella loves to hang out with anaerobes.
Right, it's a facultative anaerobe.
Yes, so it uses up the oxygen in the wound, creating the perfect anaerobic environment for other bacteria, like oral streptococci and anaerobes, to thrive. It's a synergistic infection.
And the diagnosis of Eikenella in the lab has two weird, very characteristic features.
The first is that on the agar plate, it actually pits or corrodes the surface. It looks like it's digging a little hole in the agar, hence the name corrodens.
And the second?
The second is its odor. It has a very distinct smell, like household bleach.
So bleach smell, plus a pitted agar plate, plus a history of a fist fight. That's Eikenella.
That's the triad. And one final treatment quirk. Unlike a lot of other oral flora, Eikenella is characteristically resistant to clindamycin and early cephalosporins, but it's usually susceptible to penicillin or ampicillin. You have to choose your antibiotics carefully for human bite wounds.
Good to know. And finally, the last cousin, Kingella kingae.
Kingella is the pediatric stealth pathogen. It also lives in the respiratory tract, primarily of young children.
But it doesn't cause respiratory disease. It attacks the skeletal system.
Exactly. It has emerged as a major and often under-recognized cause of septic arthritis and osteomyelitis, especially in children between the ages of 6 months and 3 or 4 years old.
Why do you call it a stealth pathogen?
Because it can be very difficult to grow in the lab, it's a slow grower and can be very fastidious. So you might have a toddler who comes in with a limp or a swollen knee, you tap the joint, and the routine gram stain and cultures come back negative.
So you might think it's not an infection.
Right. But if the lab holds the culture plates for longer, or uses specific blood culture bottles, then a few days later, Kingella will finally pop up. So you have to have a high index of suspicion.
So toddler plus a septic joint plus an initially negative culture should make you think of Kingella.
That is the classic pattern.
This has been a massive deep dive. We've gone from the drip to violin strings, from the tank with its capsule to the clenched fist with its bleach smell. Let's try to bring it all together with a summary before we hit the rapid-fire recall.
Sounds good. So for the genus Neisseria as a whole, they are gram-negative diplococci shaped like kidney beans. They are oxidase positive, and they are fastidious, meaning they're divas that need chocolate agar, CO2, and warmth.
Okay, now for the first twin.
Gonorrhoeae, the gonococcus, it metabolizes glucose only. Its key feature is what it lacks, no capsule. Its major virulence factor is its pili, which undergo antigenic variation, the face-swapping mechanism. It survives intracellularly by using porebate to block phagolysosome fusion.
And the diseases.
It causes local mucosal infections, urethritis, cervicitis, PID, and can lead to DGI or septic arthritis. Diagnosis in the lab relies on Thayer-Martin selective agar, and critically, there is no vaccine.
Perfect. Now the other twin.
And meningitidis, the meningococcus. It metabolizes maltose and glucose. Its defining feature is the polysaccharide capsule, the tank armor. The vaccines for serogroups A, C, Y, and W target this capsule sugar.
But not for B.
Not for B. The serogroup B vaccine targets surface proteins because its capsule mimics human neural tissue. Pathogenesis is driven by a massive release of LOS endotoxin, leading to septic shock and potentially Waterhouse-Friderichsen syndrome.
And lastly, the cousins.
The cousins are all about the buzzwords. Ike and Ella, think human bites, clenched fists, pitting the agar, and a bleach smell. And Kingella, think kids' knees, so septic arthritis and osteomyelitis in young children.
Fantastic summary. Are you ready to put the listener to the test?
Let's do the rapid-fire recall.
All right. I'll ask the question. I'll leave a short pause for you to answer in your head, and then our expert will break it down with the why. Question 1. A 22-year-old male presents with a purulent urethral discharge. A gram stain reveals gram-negative diplococci inside neutrophils. Which carbohydrate will this specific organism be able to oxidize in the lab?
Glucose only. The reasoning here is that the clinical picture is classic for gonorrhea. The organism is Neisseria gonorrhoeae, and the mnemonic is G for gonorrhea, G for glucose. If it were N. meningitidis, it would be able to oxidize both maltose and glucose. You can review this in Murray Chapter 23 under Physiology and Structure.
Question 2. We have effective conjugate vaccines for most serogroups of N. meningitidis, but none for N. gonorrhoeae. What is the primary genetic mechanism that makes a gonococcal vaccine so difficult to develop?
Antigenic variation of pili via gene conversion. The bacteria constantly recombine silent PLS gene segments into the active pile expression locus. This continuously changes the protein structure of the pili, creating a moving target that the immune system's antibodies cannot effectively lock onto. This is detailed in Murray Chapter 23 in the section on pili.
Question 3. A college student with fulminant meningococcaemia rapidly develops profound hypotension and shock and dies. At autopsy, both adrenal glands are found to be massively hemorrhagic. What is the eponym for this syndrome?
Waterhouse-Friderichsen syndrome. This is the specific name for the catastrophic failure of the adrenal glands due to the disseminated intravascular coagulation and bleeding induced by the overwhelming meningococcal endotoxin load. You'll find this in Murray Chapter 23 under Clinical Diseases for N. meningitidis.
Question 4. A patient has suffered from 3 separate episodes of invasive Neisseria infections over several years, a deficiency in which specific part of the immune system should be strongly suspected.
The late components of the complement system, specifically C5, C6, C7, C8, or C9. These proteins form the membrane attack complex, or MAC, which is the primary lytic mechanism for killing Neisseria species in the bloodstream. A deficiency leads to a dramatic increase in susceptibility. This is covered in Murray Chapter 23 under Pathogenesis and Immunity.
Question 5. You are attempting to culture a cervical swab on Thayer-Martin agar. Which component of the VCN antibiotic cocktail is specifically responsible for inhibiting the growth of other gram-negative enteric rods like E. coli?
Colistin, the C in VCN. Remember, vancomycin kills the gram-positives, and nystatin kills the fungi. Colistin is the agent that specifically suppresses the other gram-negative rods that would otherwise overgrow the plate. That's in Murray chapter 23 under laboratory diagnosis.
Question 6. A man is brought to the emergency department after a bar fight where he punched another person in the mouth. His hand is now infected. A culture from the wound grows a gram-negative rod that is observed to pit the agar and has a distinct bleach-like odor. What is the most likely organism?
Eikenella corrodens, the history of a clenched fist injury, or human bite, combined with the two classic lab findings pitting the agar and the bleach odor form the classic triad for Eikenella. This is in Murray chapter 23, under the section on other Neisseria.
Question 7. Why did the development of a vaccine for meningitidis serogroup B require the novel reverse vaccinology approach instead of the standard capsular conjugate method used for groups A, C, Y, and W?
Because of molecular mimicry. The group B capsule is made of polysalic acid, which is chemically identical to human fetal neural cell adhesion molecules. This makes it non-immunogenic and a dangerous target for a vaccine. The solution was to sequence the genome to find unique protein antigens instead. You can review this in Murray chapter 23 under prevention.
Question 8. What is the primary mechanism of action for the N-gonorrhoeae virulence factor that allows it to survive and even replicate inside of neutrophils?
The POR-bute protein inhibits phagolysosome fusion. PORP, a porin protein, prevents the lysosome, which is full of acid and digestive enzymes, from fusing with the phagosome containing the bacteria. This creates a protected intracellular niche for the gonococcus. This mechanism is explained in Murray chapter 23 under pathogenesis.
That wraps up our investigation into the diplococci. It's a heavy file, but these are pathogens that absolutely demand our respect.
They do. They really remind us that the line between a harmless throat bacteria and a deadly killer can be as thin as a sugar capsule or a subtle shift in a surface protein.
I want to leave you with one final thought to mull over. We spent a lot of time talking about the arms race, the bacteria changing its pilia, the host making new antibodies. But think about the carrier state of meningococcus. It lives in 10% of us, peacefully.
Right. It doesn't want to kill its host.
That's actually a failure of its strategy, because a dead host doesn't transmit the organism very well. So is it possible that the lethal meningitis and sepsis we see isn't a deliberate attack, but actually just a biological accident? A catastrophic misunderstanding between the tank and an overzealous immune system.
It's a disturbing possibility, isn't it? That all that violence might just be the collateral damage from a failed negotiation at the mucosal surface.
Thanks for listening to The Deep Dive. Keep those neurons firing.
We'll see you next time.